So the structure of the blood cells start with the erythrocytes, and these are the primary cells specialized with the transportation of oxygen and carbon dioxide throughout the body. Right? So the structure of our erythrocyte now. It is a biconcave disc shape. Right? Erythrocytes have distinct biconcave disc shapes, meaning that they have a thinner center, right, than the edges. So they resemble a donut shape without a hole. Right? They're approximately 7.5 microns in diameter, right? And 2 microns at the thickest point and 1 micron at the thinnest point. Right? The shape and the size of the erythrocyte. Okay? We should have known the biconcave shape of an erythrocyte from CSEC. Alright? The cell membrane now, we know that we have a phospholipid bilayer, right? We generally know that, right? They have membrane proteins, specifically like glycophorin, and peripheral proteins like spectrin and actin, right? So these are specific for the assistance of well, mechanical stability, right? So the spectrin helps with mechanical stability. We know that the erythrocyte doesn't have a lot of organelles or anything like that, right? So it needs these proteins in order to stay together. It increases the flexibility of the erythrocyte and the mechanical stability of the erythrocyte for the spectrin. Band 3 proteins, right, are therefore anion exchange proteins, right? So they help in the transport of bicarbonate and chloride ions. We know that hemoglobin can act as a pH buffer, right? So the band 3 proteins will assist with that. Right? But we're not going too far into that. We're ending it right there. We're not going too far into hemoglobin as a pH buffer because we're asking about its transport capabilities, right? Cytoplasm now, we know that we have hemoglobin, so this cytoplasm rather is densely packed with hemoglobin, a protein that binds to oxygen and carbon dioxide, and in some cases carbon monoxide as well, right? So the structure of hemoglobin, we have 4 polypeptide chains, right? 4 tertiary structured polypeptides, right? Different alpha and beta globulins combined, right, are associated together with their prosthetic groups, which are the heme groups, right, which are ligand chains containing iron, right, iron 3 plus, right, iron 3 plus. That's why the blood is red, because iron 3 plus is red, a red ion, right? So the beauty of chemistry is showing there. The lack of organelles as well. The mature erythrocyte lacks a nucleus, mitochondria, other organelles, everything like that. It maximizes the spacing, however, for hemoglobin and reduces oxygen consumption by the cell itself. So since it doesn't have a lot of metabolic processes occurring, it doesn't use a lot of the oxygen that it absorbs. And since it doesn't have a lot of organelles, it has a lot of space for hemoglobin. Alright? So look at how it maximizes the transport. Alright? So the function specifically, we know that it functions for oxygen transport, right? So we're gonna have molecules binding to the hemoglobin. For example, oxygen would bind to the hemoglobin in the erythrocytes, right? In the lungs, where oxygen is in high concentration, then we have oxyhemoglobin, right? That is what is formed. So the hemoglobin binds to oxygen, it forms oxyhemoglobin, which is a bright red color, right? So notice, I'm so sorry, I have to refer to chemistry, Oxygen in this case would be a stronger field ligand, therefore will change the color, right, that the complex presents. So we have brighter red. Lovely, right? So in tissues where the oxygen concentration is low, however, hemoglobin will release the oxygen, and deoxyhemoglobin will be formed now without oxygen, which is a darker red, right? So that's where we have now. Never mind, let me not continue with ligand exchange and crystal field theory. But we understand the change in color, so the brighter red when it's oxygenated, and it's a darker red when it's deoxygenated. Right? So here we have blood cells, and we're looking at the hemoglobin within it, right? And the prosthetic groups, which contain heme here. Right? So the general structure of hemoglobin. So we have carbon dioxide transport as well. So carbon dioxide binds the hemoglobin in areas which have high cellular respiration rates, and carbon-amino hemoglobin is formed, right? So carbon dioxide binds an amino group in the hemoglobin, in the globin part rather of the hemoglobin, right? So it doesn't bind to the iron per se, but in the globin part, so part of the protein. So that's why it's a carbon-amino hemoglobin. And in the conversion of the bicarbonate, now the carbon dioxide converts to bicarbonate ions by the enzyme carbonic anhydrase in erythrocytes, facilitating the transport in blood plasma, right? So remember chemistry unit 1 when we talk about the hydrogen carbonate blood buffer? Is hemoglobin cause it. Hemoglobin is doing it. It causes the creation, right, of the hydrogen carbonate, and then it goes freely in the blood and creates a blood buffer. Right? So it so looking at the fact that carbon dioxide, right, creating carbon amino hemoglobin would be one of the factors for the creation of that blood buffer. Alright? So we have that there. So pH regulation there now. Right? So it acts as a regulator, as we just spoke about the creation of the buffer system. Alright? But we're not going too far into this, we're just mentioning it here and moving on. The lifespan and turnover, the lifespan of an erythrocyte is about 120 days, right? Old and damaged erythrocytes are usually removed by macrophages in the spleen, liver, or in the bone marrow, right? And then we have erythropoiesis, right? So we have the production of new erythrocytes occurring in the bone marrow, right? Stimulated by the hormone erythropoietin, right? Produced by the kidneys in response to hypoxia. Right, so we know that the red blood cells are produced within the bone marrow, and their lifespan is around 120 days. Okay, we know the adaptation to its function is a biconcave shape. What does that mean? What does it do? It increases the area to volume ratio, right? So the flexibility as well, it allows it to move and navigate through narrow capillaries, right? The absence of organelles allow for more space, right, for hemoglobin, as well as the lack of consumption of oxygen because it doesn't have a lot of metabolism happening. And in the hemoglobin is there, high concentration of hemoglobin enables for efficient oxygen transport and release. And that is it for our erythrocytes. Any questions? We'll look at the structure and the function of the erythrocytes in depth. Any questions before we move on to the leukocytes? Moving on? Yes or no? Okay, let me give you guys a second. Ask a question, and if anybody has a question to ask, please ask
Um, sir, we haven't looked at the graph.
The Bohr effect?
Yes.
Oh, we're going to look
Yes,
at it,
sir.
man. We're going to look at it. That's the final thing in this topic that we'll look at. I'm following the syllabus.
Okay.
Because right now we're at 3.1, right? And the Bohr effect is 3.7. So we have the cardiac cycle and all that to go through, and then we touched on the Bohr effect. Alright?
Okay, sir.
Lovely, no problem. So leukocytes, now these are what we call, they're commonly known as white blood cells, but I refer to them as leukocytes because that's what they are, right? And they're a diverse group of different types of cells that play different kinds of roles within our immune system, right? By the way, my favorite module is module 3, so I cannot wait for that, right? But we're gonna get there. Right, the structure of our leukocytes, no. We have our granulocytes and our agranulocytes, right? Our granulocytes, right, contain cytoplasmic granules that are visible under the microscope. So they're granular. That's why they're granulocytes, right? They're granular cells. Granulo meaning granular and cytes meaning cells, granulocytes, right? Our neutrophils are the most abundant type of leukocyte with multi-lobed nuclei and neutral staining granules. Our eosinophils are characterized by a nuclei and eosinophilic granules that stain red orange. So what we're talking about is just different stains that we're doing. You guys will look at that differently when you guys look at blood staining, right? It's not at this level of biology though. And we have our basophils, right? They have lobed nuclei and basophilic granules that stain deep purple, right? Our agranulocytes now are the ones that are not granular. They lack visible cytoplasmic granules. So our lymphocytes, right, which have spherical nuclei that dominate the cytoplasm, right? They include our T cells, our B cells, our natural killer cells, and our monocytes now, right, which are the largest type of leukocyte with kidney-shaped nuclei. They can differentiate into macrophages, right? So we could look at that. So in module 3 comes, we hit on all of these maturation processes. All right. So here our granulocytes are neutrophil, our eosinophil, and our basophil. Look at how they're granular. We can see the visible granular components, right? And we look at the lobed nuclei, right? Clear enough, right? Clear enough. And these are agranulocytes, our lymphocytes and our monocytes. So our monocytes have the kidney-shaped nuclei. They're not a lymphocyte with just a spherical nucleus rather. Cell morphology. Leukocytes have nuclei that vary in shape and number of lobes depending on cell type, and the cytoplasm may contain granules for granulocytes and appear relatively homogeneous for agranulocytes. Right, for circulation now, the movement. Leukocytes can move between blood vessels and tissues through the process called diapedesis, right, and extravasation, right. So we have these two things. So they can actually move. See them squeezing through the basement membrane, right? They can squeeze through from the perivascular space, right? In the lymph, in the through their lymph nodes into the lymph system, right? The lymphatic system, right? Through the perivascular space into the blood, right? So we have that there. They are attracted to sites of infection or inflammation by chemical signals released by damaged tissues or pathogens, right? So we have that there. The function, the lymphocytes, no, well, the leukocytes rather, right? Phagocytosis, right? So some leukocytes such as neutrophils, eosinophils, monocytes, and macrophages engulf and destroy pathogens, cellular debris, right? And foreign particles, right? They have antigen presentation. Dendritic cells and macrophages present antigens to lymphocytes, right? Initiating immune responses. And we have lymphocyte activation, where particularly the T cells and the B cells play central roles in adaptive immunity by recognizing and responding to specific antigens. We're going to talk about that way more in depth, right, when it comes to module 3. The inflammatory response is also there as well, right? Usually this is the case of the mast cells, right? But our leukocytes, right, specifically mast cells, will actually increase the production of cytokinins, right, and regulate inflammation, immune responses, right, and what we call hemopoiesis, right. So we have vasodilation, vasoconstriction, right, permeability, right. So we're gonna have more of that occurring when we have an infection, right. So we have swelling occurring. So that's one of the other functions of our leukocytes. Tissue repair and homeostasis is also there. So macrophage activity, they remove cellular debris and initiate tissue repair processes, right? And we also have wound healing, right? So leukocytes play critical roles in wound healing and tissue regeneration by clearing any type of debris, right? So it allows the platelets to go in and work. Okay. The lifespan now, leukocytes can live anywhere but through several hours to several years, right? From a few hours to several years, right? So they have a long lifespan, right? When leukocytes are produced in the bone marrow, right, through hemopoiesis, right, and their production is regulated by cytokinins and growth factors. The adaptations now, we know the adaptations and functions, right? They have a lot of diversity, so they're tailored to different types of immunity, whether it is innate, adaptive, cell mediated, or humoral immunity. When I talk about that, those different responses, they are mobile in such a way, right? So they can actually move themselves while erythrocytes generally just flow with the blood pressure and blood direction. They're versatile, so they can adapt to different functions and responses to changing environmental cues, such as the presence of pathogens or tissue damage. Right? So those are the adaptations to their function. Alright, so that is it for our blood cells, our leukocytes, and our erythrocytes. Any questions or concerns?
Sir, someone had asked to see the red blood cell
The red blood cell. Okay,
slide.
so let me see. Which slide is it? Okay, let me check the chat real quick. Which slide was it? The hemoglobin side. Alright, so I'm seeing that one. This hemoglobin, this alone or this one? Which one? I'm gonna stop on this one. Tell me if it's the one before. The previous one, this, okay. So remember the four major globulin groups, the alpha globulins, two alpha globulins, one beta globulin, and four prosthetic groups that contain the heme, right? So different heme groups. Alright, we'll look at the structure of hemoglobin in unit 1. So it's just to jog your memory. Okay? So that is it.